And our next speaker is Zachary Walsh. Zach is a fifth year medical and PhD students at the Columbia University Vangelis College of Physician and Surgeon, and is working in Benjamin Izar Labs. Zach works with protein engineering and human genetics, and is focusing on scalable generation and discovery of variant in T cell using mainly like editing screening. And today, that talks is about his work about as I said, massively scalable assessment of variant effect in primary human T cell. And with that, I leave the stage to Zach.
Thanks so much for the introduction, and thank you to all the organizers for putting this seminar series on and for giving me the chance to speak to you all today. So I'm Zach. I'm an MD PhD student in Ben Izar's group at Columbia, and I'm going to be sharing for you some work that we recently published using CRISPR base editing screens to identify variants that can be co-opted in T-cell immunotherapies to improve their anti-tumor activity. And so to give you a brief overview, cellular therapies and really T-cell-based immunotherapies are becoming more and more common in oncology. And essentially the way these therapies work is you are harnessing a patient's own immune cells, namely T cells, to target cancer cells directly. And for example, CAR T cell therapy and tumor infiltrating lymphocyte therapy have demonstrated some really strong initial activity in patients who are otherwise very refractory to traditional chemotherapy regimens. Although these therapies have had some really exciting early efficacy, they also are kind of subjected to T-cell based modes of failure. And these can include things like T-cell exhaustion, suboptimal ability of the T-cells to actually kill the cancer cells, and poor persistence of the T-cell product itself. And as a kind of cancer immunotherapy lab, our goal is to try to figure out a way to really supercharge or armor these T cells and make them more active in this setting to overcome these resistance or these failure mechanisms. And a really interesting kind of lesson of nature, so to speak, that intrigued us is that if you look at point mutations across many different T cell critical genes, you can see that single variants in these genes can actually drive a very profound and diverse range of clinical pathologies. And this can range from anything between lymphoproliferative disorders and cancer to autoimmunity all the way to immunodeficiencies. And what this led us to suspect is that there may be some proverbial needle in a haystack variants in some of these genes that T cells use that may modulate T cell function strongly and could therefore be co-opted in order to actually enhance T cell immunotherapeutic activity. And until recently, trying to test such variants, especially at scale, was really a tall task and not one that was feasible. But this changed with the advent of CRISPR base editing, which is a technique that was originally developed in David Liu's group at the Broad. And essentially, this is a kind of extension of the CRISPR-Cas9 machinery, where by fusing an endogenous base editor protein to the Cas9 protein, you can retain the kind of guide RNA-based targeting system to a specific genetic locus. But then instead of making a double-stranded break and consequently indels and knocking out your gene, you can actually make single base conversions in a very narrow window. And therefore, you can really study variant effects. And ideally, we would like to do this directly in primary human cells to keep it as physiologically relevant and therapeutically relevant as possible. And so the first thing that we worked on was trying to figure out whether you could even do this in primary human T cells. And so we established this kind of hybrid pooled CRISPR screen compatible base editing method, where first we isolate primary human T cells from a healthy donor. We activate them, and then we use a lentivirus to deliver only the guide RNA, which is just the targeting machinery, not the editor itself. And this permanently integrates the guide into our T cells and allows us to use sequencing to look at abundance of guides after some sort of selection process. And therefore, we can look at the abundance of different variants and their influence on activity. And finally, we deliver the mRNA encoding the base editor with a transient electroporation, so it's only around temporarily long enough to make the edit of our choosing, but then is degraded and doesn't stick around, making all sorts of kind of unwanted edits across the genome. And when we use this strategy in a proof of concept experiment to install a splice site perturbation in B2M, which is a constitutively expressed T cell gene, we can see that we get very guide-specific knockout of B2M with our base editor. And this is indicated by loss of B2M only in T cells that receive the guide RNA, which is the T cells that are positive for this M turquoise fluorescent reporter. And so at this point, we've established a method that enables pooled screening, but we wanted to think really hard about what should our library design look like and what should our selections look like in order for us to get the most bang from our buck. So first, we thought about target genes, and this is just an interaction map, very high-level view of the types of pathways that we're hitting, and these are all each dot represents a gene, and these are genes that are known to play roles in T cell receptor signaling and co-stimulatory signaling that we think are likely to have a strong influence on T cell activity. And so we generated a guide library, which will ultimately translate to a variant library across roughly 110 of these T cell genes. And we next wanted to think about what should we actually select on to have a relevant readout that's going to predict for us what variants are going to support an enhanced cell therapy. And we realized that there's really not a one-size-fits-all readout for this. And so what we actually did is we took these T cells and we put them through several different selection approaches and ultimately integrated all of these readouts to give us a sense of which variants promote all of them. So among these, we have proliferation, pro-inflammatory cytokine production, and activation. And what I'm going to focus on today, just for the sake of telling a kind of concise story, is our long-term proliferation arm of the screen, where after base editing the T cells, we repeatedly stimulated them every couple of days to mimic kind of a chronic antigen stimulation that you'd see in a tumor. And we looked at which T cells had a growth and survival advantage. Before we get into the screen analysis itself, I'd like to just give you an overview of how we like to look at this data from an analysis perspective. So we can start at really a bird's eye view and look variant-wide at every variant and every gene that we target. And we can plot variant effects across the multiple human donors that we do these screens in. And that gives us higher confidence in variants that have consistent effects from donor to donor. Then if we find a gene of interest or genes of interest, we can dig down and look at whether there are clusters of variants that are having similar effects that are closely associated in the protein's primary structure. And then we can go a step further and actually look at what kind of effects these variants may have biochemically and structurally in the native protein structure. And finally, we can tie this back to clinical variant annotation, because many of the variants we actually screened have some level of ClinVar classification, whether it be likely benign, likely pathogenic, or variant of uncertain significance. So starting with the bird's eye view and looking at lens one, we looked at variants that were either consistently enriched or depleted across our screen in this long-term proliferation assay. And although the gray dots are showing you variants in every gene that we screened, what I've highlighted for you at the blue dots are variants in a gene called PIK3CD, which actually encodes for the catalytic subunit of the PI3K, PI3 kinase isoform that is highly expressed in T cells. And this gene caught our eye immediately because we were seeing many variants that were both highly enriched, suggesting a growth advantage, and highly depleted, suggesting a growth disadvantage. And this suggested to us that perturbation of PI3K can have a really strong influence on T cell behavior. And so next, we took these variants and we plotted their effects on the y-axis against the actual position of the ensuing residue change in the protein's primary structure. And we overlaid domains as well here. And what you can see is that we have several hotspots popping up throughout the gene where we have many variants. Each green dot here represents a guide that's making either one or several variants. And there are a lot of variants that have similar effects that are clustering together in primary structure. And this suggests to us that these are probably structural hotspots within the protein that we can reliably perturb to generate a phenotype. And now that we have this map on primary structure, what we really wanted to do is try to understand whether there's kind of a clear structural or biochemical reason that these effects are so consistent. And so we took the empirically solved structure of PIK3CD in complex with its regulatory subunit, which is PIK3R1. And we looked at where some of these highly enriched variants were popping up. And something that immediately stood out to us when we looked at these structures is that many of these regions where there are clusters of enriched variants are electrostatic contacts between the two subunits. And for example, what I've boxed in red here is a region of PIK3CD in green, where we have all these variants. They're all perturbing this kind of negatively charged region of the protein that's interacting with this lysine-rich positively charged interface of PIK3R1. And actually something that's known in the literature is that this specific interaction between these two subunits is inhibitory on PIK3CD activity. So we suspect that the variants generated here are releasing the brakes, so to speak, on PIK3CD's catalytic activity and thus ramping up its activity in T cells. Finally, something that we thought was really interesting is that we're able to take our variants that we screened and superimpose or overlay their variant classification in ClinVar. And just to simplify here, the red is ClinVar pathogenic variants, and the dark blue is ClinVar variants of uncertain significance. And one thing that's immediately reassuring is that we see actually ClinVar pathogenic variants scoring extremely highly in proliferation. And if you look in the literature, these variants are actually canonically associated with a PI3K gain of function syndrome, which presents in patients and primarily affects lymphocytes. But what was equally interesting is that we see these variants of uncertain significance that are right up there with them and behaving similarly, which suggests to us that we're likely having some ability to reclassify these variants as well. And indeed, if you actually look at some of these variants and you dig enough in the literature, you can see that although they're classified as variants of uncertain significance, there are reports of patients who have such variants, and early in life, they present with these features of lymphoid hyperplasia, which is very much in line with their screen, showing that they promote kind of T cell hyperproliferation. And so another angle we think of these screens is, large scale variant reclassification. But to get back to our kind of screen results, something that was really important to us was taking these results and actually validating them to increase our confidence that they're real. And the first thing that we did is we looked directly at PI3K pathway activity in T cells. And so we generated some of our top most enriched and depleted PIK3CD variants one by one in an arrayed format this time. And we found that compared to this control silent mutation that we made, AKT and S6, which are AKT pathway proteins, their phosphorylation was decreased in loss of function or depleted variants from our screen. Whereas in each of the gain of function variants that we discovered that were enriched in our proliferation screen, we see highly elevated PI3K pathway activity. And this gave us some more confidence that these are indeed variants that are increasing the pathway. Next, we looked at a more kind of immunotherapeutically relevant readout, which is production of the pro-inflammatory cytokine TNF-alpha after T cell receptor stimulation. And again, what you can see is that boxed in blue is our loss of function variant. And you can see the T cells with this variant are making less TNF-alpha after being stimulated, while conversely, any of these PIK3CD variants that we're engineering are really ramping up TNF-alpha production. But this really brings me back to the guiding, the goal of our approach, which was not just to look at variants that increase signaling or that increase cytokine, but really to see if we could identify variants which we could co-opt in antigen-specific T cells for immunotherapy to enhance their killing of tumor cells. And so we took advantage of two models that we have up and running in the lab, one of which is T cells that are engineered with a tumor epitope-specific T cell receptor targeting melanoma, and then CD19 targeting CAR T cells that recognize the CD19 antigen, which is commonly overexpressed on leukemia. And we went and we engineered some of these top hit variants from our screens in either CAR T cells, shown here, or NY-ESO T cells targeting melanoma. And here, actually, the lower the bar is on the plot, the better the tumor killing, because the y-axis is actually surviving frequency of tumor cells. And what you can appreciate is that compared to control non-targeting guide RNA edit, or compared to CTLA-4 knockout, which has previously been shown to kind of have a benefit for T-cell immunotherapy, each of our gain-of-function variants is further improving anti-leukemia activity. And now in our melanoma model, we perform similar co-cultures, and we use melanoma cells that are actually lacking CD58, which we've previously found in our group to make them highly resistant to immunotherapy. They're really difficult to kill, and actually really up until this point in the lab, we'd never seen an immunotherapy be able to clear them, even in a dish. And we co-cultured our engineered base-edited TCR T cells with these melanoma cells, and really excitingly, we found that they also had enhanced tumor killing in this setting. And what this suggested to us is that, although these variants may not be completely one size fits all for immunotherapy, they definitely seem to be broadly applicable across some of the most clinically relevant forms of T cell therapy. So I kept it a little short and sweet today, but some important takeaways that I think come out of this. One, we have this really nice platform that allows us to perform these large pooled screens in primary human cells and directly perturbing the endogenous locus. And this gives us a unique ability to really, in a physiologically relevant way, study variant effects. And we took advantage of this platform to discover single variants in critical T cell signaling genes, which we're able to use to either tune T cell activity up or down, depending on the variant. And this was, well predicted from the screen results themselves. And finally, we took some of the top scoring PI3K gain of function variants from these screens, and we used them to enhance multiple clinically relevant T cell based therapies. So this work is now available at Nature Biotech, and the QR code links to the article here. If you have any issues accessing it, feel free to also DM me and I can give you the PDF. And finally, I'd like to acknowledge really so many great contributions from members of our group and from other groups at Columbia, without whom really this work would not have been possible. It was a really high risk but exciting project, and there were a lot of people contributing to get it off the ground, and also to all of our funding sources for providing the funding for this high risk work. Yeah, with that, I'm happy to take any questions. Thanks for your attention.
Great. Thank you, Zach, for amazing talk. Now, if you have any questions regarding the talk, please use the Q&A panel. We'll read your questions to the speaker. And let's see. Okay, so I asked the first question. So in your screen, you use 2 donors, right? But potentially we have a lot of genetic background for different individuals. And how do you think the genetic background of the donors will affect the screen outcome of the variant effects?
Yes, this is a really great question. It's actually one that we think about a ton in the lab. And you're totally right that 2 donors probably gives us the ability to pull out some of the most impactful variants that are going to have penetrance, across multiple genetic backgrounds. But something we're really interested in doing going forward is not to just be able to pull out these highest activity variants in a couple donors, but try to understand how variant effects are going to have variable penetrance or be co-modulated by other variants across the genome. And so, I think what your question gets at is that we, by doing these kinds of screens in many more donors, we may be able to learn some really interesting things about variant co-regulation. And that's something we're very interested in.
Thanks. Any questions from the audience? Okay, I ask one more then. So so you target like around like 100 genes. Some of them are, I think, like probably essential, and some are like non-essential. Do you find any difference between like the gene, or like similar to like the previous talk, like the distribution of variance effects like across different genes?
Yes, very much so. So actually one thing that I didn't mention in the talk for conciseness, but a control that we included in our library was actually we used kind of a list of pan-essential quote unquote genes for human cell function that range from ribosomal biogenesis to DNA replication. And we designed a kind of sub-library of guides that were going to perturb these essential genes, either with splice site mutations or proline conversions that were likely to really screw up their structure. And what we saw with those genes over time is that the variants in those genes were highly depleted, actually, really more so than any of the other T cell signaling variants that we had in our library that were more focused on, trying to uncover gain of functions. But then when we compared, however many, like over 100 of these genes across our screen, there were definitely some where the loss of function variants seemed to have a much more pronounced effect on T cell growth and survival. So something like PIK3CD actually stood out to us, not only because of these kind of highly enriched variants, but also because when we saw proline conversions in highly structurally ordered regions of PIK3CD, those variants dropped out really heavily. So it suggested to us that this is a gene that we can look at that, the dynamic range for tuning T cell function is quite high.
Okay, great. Okay, thank you. If there are no questions, thank you everyone for joining us today. We'll have our seminar series like every month, like the first Tuesday. So I hope to see you next month.